Welcome to the last part of the course. These next 6 lectures will be on exam 4. The first 2 have to deal with you and your kidney. In other words, the process of osmoregulation. So in this lecture, we will talk about a little bit about the anatomy of the kidney, more so the cellular morphology and how each region of the kidney, specifically the nephron, which is the functional unit of the kidney, allows osmoregulation. Part 1, we're going to talk about kidney overall, the nephron overall, and 2 of the functions. In part 2, you'll hear about concentration, secretion, and the first 2 hormones of the course. Okay, osmoregulation is the prime driver of kidney function. And what osmoregulation means is we're going to regulate the salt and water balance of your blood, which means your tissue fluids as well. So here we have an active way of removing water or adding water. And of course, if you add water to your urine, you'll have a dilute urine. If you remove water in kind of a antidiuretic approach, you'll actually, by putting water back into the blood, you'll increase your blood pressure. We are going to regulate, at the same time regulating water, you're going to regulate salt balance, in particular sodium. Another salt that you will regulate is hydrogen ions, which means you're going to be regulating the pH of your blood. And finally, as we know, the other elements that flow out of the urine are various waste products. Okay, so again, we'll do a little bit of anatomy for the kidney and the urinary system. And no, you will not have to know these tissues over the next couple of slides for your exam. You'll have to know it for human anatomy, though. Okay, as the previous slide states, you have 2 kidneys in kind of the rear of your abdominal cavity. Each kidney drains by its own ureter. The ureters join and fill up the urinary bladder, and when that urge hits you, smooth muscles of the urinary bladder contract, forcing the urine through the urethra and then into that porcelain flushing facility. If you look at the structure of the kidney itself, it is divided into basically 2 halves, at least the tissue part, the renal cortex and the renal medulla, and we'll talk about that in a bit. There's also a drainage flow before it hits the ureter, and that is known as the renal calyx. Okay, again, to repeat what we talked about in the previous slide, if you look on the upper left, you'll see a cross-section of kidney. You will see the drainage entry that is known as the renal calyx. Your book calls it the minor calyx and the major calyx, but it's the renal calyx. And then you see kind of a light pink and kind of a darkish pink material that surrounds the calyx. The outer layer is the cortex. The inner layer is the medulla. So in this case, anatomy is a bit important because, as you will see in a next set of slides, the renal medulla is very important for producing and maintaining an osmotic gradient to help with water removal. If you look on the bottom part of the slide, you'll see this yellowish, curvy, noodly-like structure. That is known as the nephron. The nephron is the functional unit of the kidney. It is the nephron that we're going to spend the next, I don't know, those 22 slides and the remaining, and part 2, we're going to talk about you and your nephron. Before we go to the next slide, which again talks about the nephron, take a look at the figure on the right. Again, it's a smaller section of the cortex on the outside and the medulla on the inside, light pink and dark pink. And now the author of the book has put in the positioning of the nephron. Please note that the nephron kind of is a perpendicular, starting from the renal cortex, descending deep into the renal medulla. This is important for the functioning of the kidney. The nephron. Again, the nephron is the functional unit of the kidney. There's approximately 1 million nephrons per kidney. The nephron is, again, a series of tubes in which blood will be filtered, removed, things will be added, things will be removed, and the site of the 2 hormones that we'll talk about, site of action of the 2 hormones that we'll talk about. Again, here's the nephron. The nephron is in yellow. The nephron starts kind of in the upper left in a structure known as Bowman's capsule, which is that C, that backward C-shaped that you can look at. Nephron then continues to a curly structure known as the proximal convoluted tubules. After that, there is a long loop as the loop extends deeper and deeper into the medulla. This is known as the loop of Henle. Your book calls it the nephron loop. But really it's 2 separate structures, both structural and functional. There's a descending loop as it descends deeper and deeper into the saltier tissues of the medulla, followed by the ascending loop, which moves from the medulla back up towards the cortex. There it connects to the distal convoluted tubule, and the nephron exits into a common collecting duct, which receives filtrate, soon to be called urine, from many, many neighboring nephrons. Okay, this slide shows you some of the phenomena that go on with the nephron. And we're going to be talking about all 4 of these events highlighted in red. First of all, within Bowman's capsule, we'll talk about filtration. This is the blood filtering the plasma into the nephron. Our second stop on our tour of the nephron is at the proximal convoluted tubules, where the event known as reabsorption occurs. And this is where most of the material that has been filtered gets reabsorbed back into the blood. Number 3 is concentration, and that occurs within the loops of Henle. The last bit is secretion, and this is found within the distal convoluted tubules and the collecting ducts. And this is where material is taken from the blood and put back into the filtrate. So it's secreted into the nephron. Again, after it goes through all these features, the filtrate is then moved into the common collecting duct and travels to the renal calyx, to the ureter, to the bladder, to the urethra. Okay, again, picture on the right shows you the kidney. You'll see kind of in the kidney bean-shaped structure, kind of at 11 o'clock on the kidney clock, you'll see the positioning of a single nephron. Again, showing you that the backward C, the Bowman's capsule, the beginning of the nephron, occurs within the cortex, and the loop of Henle extends deep into the medulla, the salty medulla. Why is it salty? Take a look on the picture on the left. This again shows you kind of the nephron flowing from the proximal convoluted tubule to the distal tubule. And of course, in between is a loop of Henle. Please note how the loop of Henle extends deeper and deeper into the medulla. Also, deeper and deeper into saltier tissues. The numbers on the left are in milliosmoles, reflecting the osmolarity. The higher the number, the more salts. And when I'm talking about salts, yes, I'm talking about sodium and chloride, but I'm also talking about other salts, potassium, and thing that a chemical you don't think about as a salt, urea. To illustrate how salty these tissues are, the saltiness of the ocean is 1,000 milliosmol, as this figure clearly shows the tissues of the inner medulla, or the lower medulla, the one closest to the renal calyx, have osmolarities greater than that of the ocean. Okay, before we go to the steps, just want to show you one more picture of the nephron. Nephron here is in brown, starting with the Bowman's capsule. This time around 9 o'clock on the nephron clock. And it moves through the convoluted tubules, the loop of Henle, the distal convoluted tubules to the collecting ducts. Please note one more thing about the nephron and all nephrons. Okay. Note that there is this interesting little capillary bed that follows the nephron, especially at the loop of Henle. This capillary system is known as the vasa recta. The vasa recta is vitally important to help maintain the osmotic gradient within the medulla. We'll talk about that in part 2. Picture on the right is showing you where material is removed from the filtrate. In this case, the nephron is in blue, and put back into the blood. And you can see the blood in red paralleling the nephron. So much for morphology anatomy. Shall we talk about some physiology? Yes, I hear you. You yell at your computer screen. Okay, or your tablet screen. Okay, or your phone. Let's talk about filtration. All right. Filtration is the first physiological step of osmoregulation within the nephron. And the site of filtration, good test question, is Bowman's capsule. Okay. Now, Bowman's capsule is the first part of the nephron, and it surrounds a tangle of capillaries known as the glomerulus. So if you take your right or left hand, I don't care, open your hand wildly, take the fist of the other hand and stick it into your palm, and then with your fingers close on the fist. Congratulations! You have made a wonderful model of Bowman's capsule, which is the outstretched hand, and the glomerulus, which is your fist. Within the capillaries that make up the glomerulus, there are a bunch of holes. These are called fenestrations, and these holes allow water and small molecules to leave the blood and enter Bowman's capsule. Now, these holes are small enough that prevents blood cells, even the smallest blood cell in your body, red blood cells, way too big for these small holes, and large proteins to exit the plasma when healthy. Again, you'll see this on the next slide, which obviously shows much better than just words, but the plasma from the blood, so this is plasma plus small molecules, so salts, glucose, amino acids, and other very small molecules that are not proteins or other large molecules like drug metabolites, can easily squeeze through the holes, the fenestrae, if you are into Latin, of the glomerulus. The liquid plus the salts, or the filtrate, enters Bowman's capsule, and Bowman's capsule, with containing a cell called a podocyte, which have kind of amoeboid extensions known as pedicles. These have slits, and the pedicles can cover the slits or not cover the slits. And so Bowman's capsule, via these podocytes, can also regulate the flow of material into the nephron, Bowman's capsule. Okay, again, this is basically showing you exactly what I described in the previous slide. So let's take a look. On the left is the cartoon drawing of the glomerulus surrounded by Bowman's capsule. Okay. You see that the holes of the capillary, the fenestrae, allow material to move through and then eventually enter Bowman's capsule. But even within Bowman's capsule, there are ways to regulate how much plasma filtrate moves in. And that is done with the help of the podocyte, covering or not covering the various slits. If you take a look on the right, you will see a really nice transmission electron micrograph picture of the same process. So this is a real photo. At the bottom, you'll see what looks like this big circular gray blob. That's the red blood cell, the erythrocyte. You'll also see a hole or fenestra in which material can move in, and the slits that make up the Bowman's capsule, which can be regulated by the podocyte, or what your book calls the foot processes. Okay, so again, the combination of the fenestrae, the holes in the capillaries, and the slits within Bowman's capsule help regulate how much water moves in and how much salts and others move in. Both provide a huge barrier for the filtration, the moving of blood cells and large molecules like proteins. Now, if you have protein in your urine or blood cells within the urine, that is usually a sign of some sort of kidney problem like infection, kidney stones, cancer, etc. Now, the filtrate and the amount of filtrate that gets through the glomerulus into Bowman's capsule is highly dependent upon blood pressure, or kidney physiologists don't care about blood pressure because they're worried about hydrostatic pressure of the blood. Yeah, it's the same thing. There are other different types of pressures that help add things in, like colloidal pressure for the amount of proteins in there, ionic pressure for salts and things like that, and there's a net amount of pressure that ensures that plasma moves from glomerulus into the nephron, Bowman's capsule. Okay, all right, to sum up filtration, the plasma of the blood minus the proteins is filtered into the nephron. Okay, again, this can be regulated primarily by the podocytes in Bowman's capsule, but on average, the amount that gets filtered every minute is a little over 100 mils per minute. If you do the math, you'll find out that the total blood volume is filtered every 40 minutes or so. And every day, approximately 180 to 200 liters are filtered through both kidneys every day of your life, assuming you have healthy kidneys. But wait, I hear you claim, depending upon the size of you, you have between 4 and 6 liters of blood. Am I losing all that blood? Remember, this is just getting filtered. Most of the material, water, salts, sugars, amino acids, etc., get reabsorbed, thankfully. Shall we talk about the second step of kidney function, known as reabsorption? So, again, you had this massive amount of plasma being filtered in Bowman's capsule. In the next part of the nephron, the proximal convoluted tubules, about 70 to 75% of what got filtered originally in Bowman's capsule now gets reabsorbed from the filtrate, the nephron, back into the blood. So 180 to 200 liters of water is filtered per day, but you only excrete 1 to 2 liters per day. So 99% of all the material that you got that was filtered in Bowman's capsule is returned to the blood. Okay. Obviously, this amount will increase whether or not you are hydrated or dehydrated when you're hiking or enjoying yourself at the bar with all those shots of tequila. Okay, so again, this is just showing you the initial filtration within Bowman's capsule and the reabsorption of most of the material in the proximal convoluted tubule. Reabsorption occurs with a couple of interesting processes. The first process is to pump out sodium. So there are sodium pumps within the proximal convoluted tubule. And this pumped out into the blood sets up a higher salt concentration in the blood that helps drive osmosis and allows for water reabsorption, removal of water from the filtrate. Okay. So we had the active transport of sodium, the passive transport of water, and as water moves in to the area, other salts and perhaps other materials, chemicals, will also diffuse, again passive, back into the blood as well. This slide merely showing you the active transport of sodium from one side of the nephron to the other, proximal convoluted tubules. And then From there, it's picked up by the capillaries that surround the proximal convoluted tubule. Again, this is merely a cartoon showing you what is occurring within the proximal convoluted tubule using reabsorption. Salts aren't the only chemicals that are filtered. There are also small molecules such as glucose, blood sugar, and amino acids. Both glucose and amino acids are also reabsorbed into the proximal convoluted tubule using a mixture of simple diffusion or some sort of pump that will allow these chemicals to get reabsorbed. Okay, this is the last slide for You and Your Kidney Part 1. So let's kind of sum up the first two steps of the physiology of the nephron. So, blood plasma is filtered in Bowman's capsule. It then travels, it's known as the filtrate, and travels to the next part of the nephron, the proximal convoluted tubule. Here, approximately 70 to 75% of all the salts and water are reabsorbed here. They don't go any further. So what goes further, which of course is going to be in the second part of the lecture, is only about 20% of the water and salt required to get balanced. And so if you think about it, the next part, the loop of Henle and the stage of concentration, they're only dealing with a small amount of water and salts to deal with rather than the entire plasma. So the majority of water and sodium is going to be reabsorbed through the loop of Henle. The water will get reabsorbed in the descending loop. Sodium and chloride will be removed in the ascending loop. Okay. At the distal convoluted tubule, there can be other materials removed, such as water. So it's kind of, at this stage, it's kind of fine-tuning. And this is under hormonal control with the hormone ADH, antidiuretic hormone. And we'll talk about that also on part 2.